Hi, good day. My name is Janice Macayan from BSN 1A, and today we will tackle about enzymes. So, what is enzyme? Enzymes is an organic compound that act as a catalyst for biochemical reaction. So, enzyme, it means that they help in breaking down example foods that we intake for example, those digestive enzymes like pepsin, they are the one that help breaking down foods that that made from cellulose or starch, so that uh, it can easily be digested by our stomach and intestines. It also came from Greek word which means and came from in enzyme means yeast. So enzyme is a yeast. So most of the enzymes are globular proteins. And it is undergo denaturation. So nag 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 denature gihap an enzyme. In addition to the history of an enzymes, um, dati it was just understood a simple understanding because di ba usually enzymes, oh no, yeast, we use yeast for making bread and production of beverages. uh, in bread, they use yeast to produce carbon dioxide to for the bread to rise up, while in production of beverages, fermentation occurs wherein it's break down those basta. For examples ba and mga grapes, di ba ginihimo yung wine. So that's it. So an enzymes na undergo denaturation due to alteration of the pH, temperature, um, yeah. So, for examples, ang temperature, hayuman. So, pagkataas si tiranat, like, sinanta 40 degrees Celsius. So, nagkakadan alteration it aton body, diba? So, ito, it's some of the enzyme functions. Bakit sugan? Kung bagaan enzyme, so, sa sobra ka pa, so, diri lugod hiya nakakag, um, bagat, ano to, bagat, na-alter an iya functions. So, the next is the structures of an enzyme. So, may tayo si nga simple enzymes and conjugated enzymes. Wherein and simple enzymes composed only um, composed only of protein amino acid chain. So, usa lahiya nga chain. While unconjugated enzymes has a non-protein part in addition to protein part. Tapos, no catalytic properties siya. So, ano ito nga mga non-protein part? Tapos, protein part. So, ang protein part, amin yung tinatawag nga apoenzymes. So, an apoenzymes. While a non-protein part, and cofactor. So, ini nga apoenzymes, enzyme ini nga kinanglan niya hin cofactor para iya mag-function properly. So, tas adiliwat nga holo enzyme, apoenzyme plus cofactor is holo enzymes. So, next is the nomenclature and classification of an enzyme. So, nomenclature, enzymes, they are named by using a system that attempts to provide information about the function of the enzyme. So, an enzyme diri hiya ginngaranan hiya truhan function dirihan iya structure. So, true functions. So, amon. So, kumbaga, ang type of reaction, catalyze, and substrate at identify the focal points for the nomenclature. So, amon. So, ano niya sinistring nila nga substrate? Substrate is the reactant in an enzyme catalyzed reaction. It's a substance on which an enzyme acts. So, any nga substrate, amin it can be break down heat enzymes. So, pag specific line niya para nga active site enzymes. So, it is what may niya important aspect of naming. So, nga tanan nga may mga suffix ase or ase ngan in are enzymes. Ini nga in like pepsin, trypsin, ansyahan kasi ini nga mag-study iya. So, ini amin and dati nila nga pag-name an in an era suffix. So, usually yan na it mga enzymes are mas madali na ma-remember through the suffixes ase and in. Tapos, the next is type of reaction catalyzed by an enzyme is after noted with a prefix. So, for example, oxidase and hydrolase. So, kung kung nag-undergo oxidation, so, an enzymes nga gagamiton, oxidase. So, tapos, kung water liwat, syempre, hydromelt water, so, hydrolase. So, the next is the identifier of the substrate is often noted in addition to the type of reaction. So, for example, glucose oxidase, pyruvate carboxylase, and succinate dehydrogenase. So, yun. So, the next is the models of an enzyme action. So, enzyme active site. So, ini active sites, only a small portion of an enzyme molecules. So, it is three-dimensional. Tapos, three-dimensional. It is a crevice-like. Crevice-like. So, para dili kita mag-imagine nun, I'm gonna draw it. Oh no, I didn't know how to draw. Yun! So, inihiya siya ni its active site, heat, enzyme. So, enzyme. active site. So, ang substrate, kung same hiyahin structures, it will be fitted here in the enzyme active site, and then pag kuan mabe-breakdown na yun. Yeah. So, ang next, muna to ang enzyme substrate complex. So, amay niyaan the intermediate reaction process that is formed when a substrate bind to the active sites of an enzyme. So, na yung substrate complex na yapag and substrate nag-bind na hiya nga diha active sites han enzyme. So, ang hitsura lawat han substrate. Sige. Sugad. Tsurahan substrate. So, pag mag-combine hira, sige, combine na. God. So, asya na ito. Magiging itsura pag nag-combine, nag-bind na hira. Tapos, after, magbe-breakdown. Tapos, ato nga akong pag ang substrate and enzymes combined, bind together, tatawagan natin na lock and key model. So, asya na to. So, dapat shift fit ngan induce fit model. Amo na to. So, the next is the enzyme specificity. So, may mga specificity and enzyme. So, the first is absolute specificity. Absolute specificity. Catalyze only one substrate. Sala nga substrate. Examples, uris. Sala. So, the next is stereochemical. Distinguish between the stereoisomers. So, and chirality niya. So, kung ano laan fit nga to asya it mabind nga tohan koanhan enzyme active sites and enzymes. So the next is group specificity. Group specificity structurally similar compound that has the same functional group. So mga compound nga para pareho it function. So nga tera dito same bind. For example, carboxy 50 days. Then the next is the linkage specificity. Linkage specificity involves a particular type of bond. So for example, mga peptide bound, specific, and linkage siya. So, mga ester linkages, mga amide linkages ba to? Tapos, next is the factors affect enzymes activity. So, amin nira naga-alter han function han enzyme RNA activity. So, the next is temperature. So, temperature, just like what I said earlier, that too much high or overheating enzyme or yeast can cause denaturization. So, amo to. Kumbaga, amin kung sa ganang structures, di ba, usually, di ba, nasa ring globular and most enzymes are globular. So, kung sugad, nalulus siya, so, dili na yan nakaka-function yung maupay. Or, nasya na. Then, pero may ada mga microorganism, like archaea that prefer to live in hot environment. So the next is the pH. So pH is one that these are located in the active sites. So pag alter ang pH, so malulus ang catalytic activity. So diri niya makakatalyze ang substrate. So so ingon ta, for example, ang digestive enzyme na pepsin and trypsin. So ang substrate concentration liwat. So concentration of an enzyme is kept constant. Concentration of the substrate is increased. The enzyme activity pattern is obtained. So, magsa-saturation curve. So, kung damo in substrate nga nakukuan, so, an activity had enzyme, so, na-increase. So, an enzyme concentration liwat. So, iniliwat so gad kung diri an enzyme not consumed in the reaction di catalyze. So, kumbaga nagsubra kira. So, diri. The cell usually kept the number of enzyme low compared to with the number of substrate molecules. The concentration of substrate in a reaction is much higher than that of the enzymes. So the next is enzymes inhibition. So enzymes inhibitions are substances that slows or stops the normal catalyst function of an enzyme activity by binding to it. So may dining here 3 modes. The first is the reversible competitive inhibition. So in a reversible competitive inhibition, so a molecule that sufficiently resembles of an enzyme substrate in a shape and change distribution that it can compete with the substrate for occupancy of the enzyme's active sites, causing decreased enzyme activity. So an reversible competitive inhibition, so as siya ni a substance that has the same feature, the same feature like the specific substrate that need to bind in the active site of an enzyme. So, the drawing.
Baga feeling ko 6, 7, or 8. Kaya amo takin binasa talaga medyo na-cancer ako. Enumeration. Again, multiple choice.
So, ang competitive inhibition,
Mga A, B, C, D nga.
sample na ini, adi an active site, tapos, adi an, normal substrate, tapos, adi an, asya ni an competitive inhibitor. So, they have the same feature like the normal substrate that will bind here in the active site. tapos so ini nga competitive inhibitor hiyaan makombine nga dihan active site which cause to decrease in enzyme activity. So So next is the reversible non-competitive competitive inhibition. So under reversible non-competitive inhibition so diba ama the normal substrate. So atas adian active adian enzyme. So ang mangyayari and substrate it can bind to the active site but ma-alter and function or diri makakatalyze an enzyme and substrate because of the reversible non-competitive inhibition, because it will be located here at the bottom of the enzymes. So for example, so for example, amo ini so di ba amo di ang normal nga enzyme. So pag may ada non-competitive competitive substrate non-competitive substance that inhibit the function of an enzyme, so it will be located here at the bottom. Alright, so that a shiny and substance tapos pero ang substrate makakabind hiya nga di pero asya to diri makakatalyze han enzyme and substrate because of the presence of this substance that altered its function. So the next is the irreversible enzyme inhibition. So ini nga enzyme ini nga substance liwat. It is a molecule that forms a covalent bond to a part of the active site that permanently preventing such a pang-occupying it. So, iniluwat yung irreversible. Kumbaga, ang substrate, dili na talaga hiya makaka-combine nga diha active site kay may is covalent bond nga strong covalent bond nga ma-combine nga siya dun nga di rin na nga. So for examples, an irreversible good kumbaga may daba makakaulang talaga nga magiging maga-alter han binding site han substrate and enzymes. So the next is the regulation of enzymes activity. So the first is in the cell. So large amounts of enzymes. So large amounts of enzymes decreases substrate concentration. So kung damo ang enzymes, tapos low ang substrate concentration, kung damo ang enzyme, tapos low ang substrate concentration, so kung low ang naabot nga mga nga kung gutailan ang substrate, so bagat wasting time or wasting of energy hiya para ha enzymes. So ang mangyayari bagat igta-turn off nila ang activity han enzymes, bagat igdi-deactivate bagat sagad. So the other product of enzyme concentration reaction is plentifully wet. Kung damo liwat it enzymes tapos kung damo liwat it enzyme tapos so iniliwat nga cell product of enzyme concentration reaction is plentiful kung sugad damo-damoan na an product han usa nga han enzyme tapos diri naman niya kinahanglanan specific organ nga dapat kundiin hiya para nga to, so bagat waste time of energy gyapon niya para ha enzyme kay kumbaga nagsusubra naman so diri gagamiton. So the next is the allosteric enzyme. So what are allosteric enzymes? Allosteric enzymes are two or more protein chains and two kinds of binding sites, substrate and regulator. So liwat, so kung imagine nat enzyme, substrate, tas may the regulator. Pero itong regulator, may two types ito, negative and positive. So, what is the negative and positive regulator? So, the positive regulator, positive ano kita, para happy. Positive. So, amo di an enzyme, tas amo di an substrate. So, ang positive regulator, positive regulator, adi ano, tapos sige unahon natin negative ani kita ha, negative regulator. So amo di an substrate, amo di an enzyme. So So, ano mangyayari? Because negative regulator, a negative regulator, sorry, pag may ada, oh no, pagsugad niya din, pag-combine hini nga negative regulator, mako-close ini niya, so ang substrate diri niya makaka-combine nga di sugad, na gets. So unpositively what, sigunta, di ba na close ini, sigunta na close, sugad. So, ang mangyayari, ang mangyayarihan positive regulator, pag-combine niya nga dito nari. Pag-combine han positive regulator nga di, magkakada na inactive site usa nga enzyme. So, kumbaga, masugad niya. Oh, no. So, okay. So, kumbaga, makaka-bind na ang substrate to the active sites of the enzymes. So, amod to an positive nga negative regulator. So the next is the feedback control. So, feedback control is a process in which activation or inhibition of the first reaction in a reaction sequence is controlled by a product of reaction sequence. So the next is the proteolytic enzymes and zymogens. So proteolytic enzymes are produced by an active form, then when needed are converted to active forms. So zymogens are the inactive form. So proteolytic enzymes are produced by zymogens, and then they are converted into active form if they are already needed. For example, are those digestive and blood clotting enzymes. So the next is antibiotics that inhibits enzyme activity. Antibiotics are substances that kill or inhibit the growth of microorganisms. So antibiotics have two families, the sulfa drugs and penicillin. So sulfa drugs are usually used for body infections such as gastritis or and many more. Then the next is penicillin that are used usually for more serious conditions. So penicillin, it is a widely used antibiotics, and it was accidentally discovered by a scientist. So it inhibit the transpeptidase. So if you can remember the cell wall of one bacterial cell, which is the peptidoglycan. So penicillin are the one that used by doctors or prescribed by the doctors in order to kill the specific bacteria that causing the disease. So it so what to so ang gibubuhatan penicillin, diba, enzymes. So dito han active site han enzyme han bacterial cell. So they inhibit the active site so that the substrate of the specific bacterial cell will not be able to bind into that and into that enzyme active site so that asya na to. So penicillin has two unique action, but it depends on to enzyme activation. So the first is structural similarity to enzymes natural substrate. So penicillin, they are usually the same structure with the same structure of substrate, where they are going to inhibit or where they are particularly need or treat a disease. So the next is the irreversible inhibition. So diba an irreversible inhibition, they combine in a they make a covalent bond, a strong bond, so that that specific substrate won't be able to bind to that and specific enzyme active site. So penicillinase. So penicillinase are enzyme produced by a bacteria so that they can they can fight the penicillin. So uh, amato. So the next is Cepro. So Cepro are effective antibiotic against bacterial infection of different part of the body. So medical uses of enzyme. So enzyme, pag there's the presence of enzymes in the blood, so it means that there is a possible tissue damage. And so for examples an BUN, so and blood urea and nitrogen. So ang to look, actually it bibili an ammonia. So to look for it in a lab result, in a laboratory examination, usually the doctors will order the BUN. So in the presence of urea, so pag an urea is increase, so there is in the treatment liwat. So for examples, may the heart attack. So the tissue plasminogen activator or the TPA are the one that gina-activate. So ini nga tissue plasminogen activator at iya hini function is to remove the blood clots in the heart so that the blood will flow ang tawag ito in a good way bagat diretso ba, so that the heart can pump and supply other parts of the body as